In this class, we are going to discuss cell suspension culture. Cell suspension culture can be defined as growing of cells, which includes growing of single cell, that is culture of single cell or aggregates of cells in vitro in liquid medium is called as cell suspension culture. Now let us understand the meaning. Growing or culture of a single cell or group of cells in vitro. In vitro means inside the lab using liquid medium. It's called as cell suspension culture. We have studied various medium. MS medium is preferably used because it is a liquid medium. We have studied various other medium like Gambort medium, NIST medium, Weiss medium, agar-agar and so on. But in cell suspension culture we use preferably the liquid medium, so MS medium is selected. Now, how this cell suspension is prepared? It is prepared by transferring a portion of the callus in the liquid medium and agitated using rotary shaker instrument. As a result, the cells from the callus are separated and it is used in cell suspension culture. To understand this, let us see a simple diagram. Now, this is the plant, the selected plant from which we are going to take the tissue. Now, removing the tissues from a selected plant is called as what? Yes, it is called as explant. Now, I am going to take the tissues from the leaf. So, this is called as explant. Explant can be defined as the removal of tissue from the selected plant. When placed in a suitable medium, it will regenerate an entire plant. So, the explant I am taking. Explant is nothing but a group of tissues I am taking, and this tissue which I have taken from this selected plant is placed in a suitable medium. So, I am placing it in a suitable medium, preferably this liquid medium. We can take it as MS medium. Now, when this is the explant. Now, when all the conditions are favorable inside the plant tissue culture laboratory, say for example the pH, temperature, light intensity and relative humidity and all the other conditions are favorable, there is every chance that this explant will develop into a structure called callus. Now, as a result, you will be getting, so this is the medium and this medium will be having a ball of cells. This is called as callus. Now, how will you define a callus? Callus is an unorganized mass of undifferentiated tissue. So, a ball or mass of tissues is produced when all the conditions are favorable. Now, what I am going to do in cell suspension culture, a portion of this callus is separated. So, I am going to take half of this callus. I am going to keep it in another test tube. So, placing it again in the liquid medium. So, I am taking the portion of this callus, keeping it in the liquid medium. Say again it is an MS medium. Afterwards, I am going to keep this test tube in an agitator, that is called as a rotary shaker instrument. This is called as a rotary shaker instrument. Now, as a result, this test tube with a portion of the callus, now let us mark this as callus for our understanding. Now, this callus will get agitated. So, what will happen? The cells that are found in the callus will get separated and these cells can be used for cell suspension culture. So, as a result, I will be getting many cells. So, I can take all these cells in a different test tubes and placed in a liquid medium. So, I am going to place all the cells that I got as a result of the separation of this callus. So, I am taking these cells can be used in cell suspension culture. Now, the cell suspension culture play a very important role in the production of secondary metabolites. What are called as secondary metabolites? The example for secondary metabolites are alkaloids, flavonoids, terpenoids, phenolic compounds, then recombinant protein. All these are examples for secondary metabolites. But what is the meaning for secondary metabolites? Secondary metabolites are chemical substances produced as a byproduct as a result of cell metabolism. That means plants will undergo various metabolic activities. As a result of these metabolic activities, it releases a byproduct, and that is called a secondary metabolite. And is there any use for the plant? No, secondary metabolites doesn't have any role in the direct growth and the development of the plant, but it has got lot of use for the human being. So we are interested in extracting these secondary metabolites for human use. For example, Catharanthus roseus. So we are taking one example. Catharanthus roseus is the name of the plant. The common name is Vinca rosea. So Vinca rosea is a common name. It is a medicinal plant, but it is grown in garden for its ornamental value. This plant will produce an indole alkaloid called vincristin. Vincristin, which is used in the treatment of cancer. That means this vincristin has got anti-cancerous property. So which is used in the treatment of cancer. So we can write this anti-cancerous property. Cancerous agent or carcinogenic agents. Now this is the alkaloid produced from this plant. Our interest is to produce this alkaloid more in number. Now how will you produce this alkaloid more in number? We have to use bioreactor. Now we know this plant will produce this alkaloid. Now I have to take the alkaloids from the plant cell and I have to put it in the bioreactor. Let us see what is this bioreactor is all about. Now say we can take this example also. All these cells, now if it is Catharanthus roseus, this I have isolated all the cells. So all the cells will produce an alkaloid which shows anti-cancerous property. Now what I am going to do to increase the production of this alkaloid, I have to place all these cells into a bioreactor. Now what is a bioreactor? Bioreactor can otherwise called as it is a vessel or a container or a fermenter. It will be like a small machine where it is like a small machine. Now this is a bioreactor. Now I have to place it is designed in such a way it have a conducive environment for the growth of microorganism, living plant cells or the derivatives of plants like enzymes or raw materials of plants can be put inside, and we will get the final product of our interest. Say for example, you can understand this with another example. There are few green algae like Chlamydomonas, Spirulina, Chlorella. Now say you take Spirulina. Spirulina is a blue-green algae which grows in the deep sea. Now the spirulina is rich in protein. 70 to 80% of the spirulina contains dietary protein. Now spirulina cells should be taken and you have to place the cells into the bioreactor. Now what we will get? We will get the desired product. What is our product? The protein. Now we can extract the protein from the bioreactor and we can make that protein in the form of tablets, capsules or powder, and we can take as a human supplement. This is how the secondary metabolites can be increased. Now latest methods like bio transformation These are the latest or the strategies: biotransformation, elicitation, immobilization, are the techniques which can be used in cell suspension culture to increase the production of secondary metabolites. Now let us see few examples where industrially used secondary metabolites. The first one is digoxin. So digoxin is it is a secondary metabolite. This digoxin is taken from the plant source Digitalis purpurea. This is the plant source. Plant source is nothing but from this plant we are taking this secondary metabolite digoxin. Now what is the use? Now uses. Now uses is, it is used in the treatment of heart disease. That is, it is used as a cardiac tonic. This is one. So this digoxin is a secondary metabolite obtained from Digitalis purpurea, which is used in the treatment of heart diseases. That is, it is used as cardiac tonic. Now next one is codeine. Codeine is a secondary metabolite obtained from Papaver somniferum, which is used as analgesic. That means painkiller. It can be used as a painkiller. So codeine is the secondary metabolite obtained from the plant source Papaver somniferum, uses as analgesic. Then another one, capsaicin. Capsaicin is the name of the secondary metabolite obtained from Capsicum anum, which is used in the treatment of rheumatic pain treatment. That is, in rheumatoid arthritis. That means pains in the joints. Now one more example, it is vincristine, which we saw already. Vincristine is the name of the alkaloid obtained from Catharanthus rosea, which is used as an anti-cancerous or anti-cancerous property. Now the last one is quinine. Quinine is the name of the secondary metabolite obtained from Cinchona officinalis. We can get this quinine either from Cinchona officinalis or Cinchona calicea from two different plants, and it is used as an anti-malarial drug. Now this is how secondary metabolites can be extracted using cell suspension culture. So cell suspension culture play a very important role in the production of secondary metabolites, which you have to keep it in the mind. Now the next topic is plant regeneration pathway. The next topic is plant regeneration pathway. Now as we all know, explant play a very important role in the regeneration of an entire plant. Now this explant can be regenerated into an entire plant either by two important processes. One is somatic embryogenesis, another one is organogenesis. Let us see how an explant is developed into an entire plant by somatic embryogenesis and organogenesis. Now first we will take this somatic embryogenesis first. Somatic embryogenesis is nothing but development of the embryo directly from the callus is called as somatic embryos, and the process of production of somatic embryo is called as somatic embryogenesis. Now first one is the explant. Now this is the explant. When this explant is placed in the nutrient medium, it can be any medium, MS medium, Gambox medium, Nitsch medium, white medium, or any medium can be selected. Now you have to place this explant into this medium. Now as a result, a callus will be formed. If all the conditions are favorable, only when the conditions are favorable, we know all this plant tissue culture, we have studied the basic plant tissue culture technique, what are the conditions. When all the conditions are favorable, there is every chance that this explant will develop into a mass of cell or ball of cell called as callus. Now callus is an unorganized mass of undifferentiated tissue. So from the callus, if an embryo is developed directly, so from this callus an embryo will be developed. So this is the embryo. This embryo is called as somatic embryos. You should not call it as an embryo, but you should call it as a somatic embryo. Now why are we calling it as somatic embryos? Because real embryos will be produced as a result of fusion of the male and the female gamete, which we have already studied in the first lesson. Now here we are producing an embryo from an explant taken from the root, stem and leaf. So that is why we should call it as somatic embryos, or otherwise called as somoclonal embryos, or otherwise it should be called as embryoids. Now what is the use or what is the application of somatic embryogenesis? The first application is somatic embryogenesis or somatic embryo produced will result in the formation of a plantlet. So from this embryo a plantlet will be produced. This plantlet is produced inside the test tube, and these plantlets should be transferred first into the greenhouse and then into the field condition. That process is called as hardening. So after hardening, this plant can be a plant. Now this is plantlet. Now this plantlets will be developed into an entire plant by hardening technique. This is one, that is somatic embryos can be developed into a plant. That is the first important point. The second one is once the somatic embryos are formed, you can encapsulate it. Suppose, say for example, this is the somatic embryos that is formed. Over it, you can encapsulate. So that means you are encapsulating this embryo. Now this is the embryo inside and this is the encapsulation. How will you encapsulate it? You are encapsulating with calcium alginate or agarose gel. Either this embryo can be covered with calcium alginate or agarose gel. So once you make the embryo covered with calcium alginate or agarose gel, you should call this as synthetic seeds. So this is called as syn seeds or synthetic seeds. So synthetic seeds play an important role in the region of plant, not now, in future. Now the third point is somatic embryogenesis has been practiced in many plants like Allium sativum, that is in garlic, Hodium vulgate, that is barley, Oryza sativa, that is in rice, and in Zea mays. Not only in these plants, but they have practiced in almost all the plants. Somatic embryogenesis was very successful in all the other plants. The next one is, the next topic is organogenesis. Now the next one is organogenesis. Now organogenesis is nothing but development of organs like root and shoot from the callus is called as organogenesis. Now say for example this is the callus. Now this callus after differentiation, so it will undergo differentiation, and as a result of this callus undergoing differentiation, which results in the formation of two important organs. One is root, another one is shoot. The formation of the root is called as rhizogenesis. And the formation of shoot is called as calogenesis. And as a result, a new plantlets will be produced. So a plantlet will be produced, and this plantlet will develop into a plant. So it is nothing but now callus. Now callus is an unorganized mass of undifferentiated tissue. Afterwards, once the callus undergo differentiation, from this a root will be developed and a shoot will be developed. So an embryo with root and shoot is called as organogenesis. That is nothing but formation of organs from the callus is called as formation of organs like root and shoot from the callus is called as organogenesis. So in this class we have understood cell suspension culture, somatic embryogenesis and organogenesis in detail. Please refer your textbook and learn these things properly. Thank you.